Welcome back to the SANS Science Experience, where we explore the most exciting new research papers and preprints just for you. Hey, if you enjoy these deep dives, please do take a second to like this video and subscribe. It genuinely helps support the SANS Science Experience. So imagine if we could actually slow down, maybe even partly reverse, aging across the whole body. Sounds like pure sci-fi, right?
It really does, but there's this fascinating new paper in Cell this year that's giving us a real glimpse of that possibility.
Yeah, I saw that one. And what makes it stand out, I think, is the approach.
Yeah.
Using genetically engineered human cells.
Right.
And then seeing what happens in primates, specifically looking at aging markers. So our mission today is to kind of unpack this research, get to the core findings, and figure out why it matters to you.
Absolutely. And the key player here is a special type of cell they created. Senescence-resistant human mesenchymal progenitor cells. Let's just call them SRCs. It's easier.
SRCs.
Okay.
And these are basically modified adult stem cells.
That's right. A modified version of mesenchymal progenitor cells designed to be, well, tougher.
Okay, so how do you make a cell senescence-resistant? What's the engineering trick?
Well, they did a very specific genetic modification, a mutation in a gene called FOXO3.
FOXO3, okay. Known for its role in stress response and aging.
Exactly. The tweak was designed to make these SRCs more resilient to stress, less likely to become senescent.
Yeah.
You know, that state where cells stop dividing and can cause inflammation.
And they confirmed this worked in the lab first.
Oh, yeah. In lab tests, these SRCs performed better, less senescence, better proliferation compared to normal cells, live longer, divided more effectively.
So you've got these supercells. Then comes the big test. Aged primates. Tell us about that setup.
They used aged cynomolgus monkeys around 19 to 23 years old. Biologically, that's pretty similar to humans in their, say, late 50s to late 60s.
We have a relevant model then.
Yeah.
The experiment itself, how long did it run?
The monkeys got infusions every 2 weeks for 44 weeks.
Wow, that's quite a long study. Nearly a year.
It is. It's roughly like looking at 3 years of effects in human terms. They had different groups. One got saline, a control group.
That's standard.
Another got normal unmodified cells. And then the key group got these engineered SRCs.
Okay, crucial question before we get to the results. Safety. Any red flags?
That was actually a really positive finding. They reported minimal adverse health effects, no significant changes in immune markers, and the monkeys getting either the normal cells or the engineered SRCs.
That's good news. And concerns about, say, tumor formation with cell therapies.
They addressed that too. Separate studies in mice, even with high doses, didn't show tumor development. So the safety profile looked pretty promising.
All right, let's get to the really exciting part. What did the SRCs do to the aging process in these monkeys?
This is where it gets fascinating. The effects were systemic, widespread. They looked at 61 different tissues across 10 organ systems.
61 tissues.
Yeah. And the SRCs seem to counteract age-related changes in, well, many of them.
So not just one specific area, but like a broad rejuvenation effect. Can you give us some examples?
Definitely. Take bone health. They measured less age-related periodontal bone loss, around the teeth. And the structure of the inner spongy bone, the trabecular bone, also showed improvement.
Okay, bones. What else?
Blood rejuvenation was another one. They saw shifts in gene expression patterns in blood immune cells, the PBMCs, moving away from the aged profile.
And you mentioned inflammation earlier, which is such a key factor in aging.
Right. The SRC treatment significantly reduced chronic inflammation across multiple organs. Attenuating that widespread low-grade inflammation is, well, it's a really big deal for healthy aging.
Were there any areas where the results were particularly striking?
I'd say the brain and the reproductive system effects really stood out.
The brain, what happened there?
Improved performance in memory tasks, for one, suggesting maybe a slowing of cognitive decline. They also saw enhanced functional connectivity in the hippocampus.
The memory center.
Precisely. And signs that age-related thinning of the myelin sheath, the insulation around nerve fibers, was being lessened.
That's potentially huge for brain health. And the reproductive system.
Also quite remarkable. The SRCs seemed to significantly rejuvenate reproductive organs. It was particularly pronounced in the ovaries. Get this. They estimated a reduction in the biological age of the egg cells, the oocytes, by over 5 years.
5 years? Compared to what?
Compared to the control group, essentially. The monkeys treated with the normal cells only showed about a 3-year reduction, so the engineered cells had a much stronger effect there.
Incredible. So how are these few infused cells having such wide-ranging effects across the entire body? What's the thinking on the mechanism?
The researchers propose it's largely down to exosomes.
Ah, the little messenger packages cells release.
Exactly. The idea is that these SRCs are releasing exosomes packed with beneficial molecules, and these exosomes travel through the body, delivering these geroprotective or anti-aging signals.
So it's the cargo inside these exosomes that's doing the heavy lifting. What kind of stuff is in there?
They found the SRC-derived exosomes were rich in proteins like antioxidants, anti-inflammatory factors, and also certain metabolites like spermine, which has known rejuvenating properties.
Did they check if just the exosomes without the cells could work?
They did. They ran tests in mice and also on human cells in culture using just the exosomes isolated from the SRCs. And those experiments showed that the exosomes alone could reduce aging markers and improve tissue health indicators. It supports the idea that these packages can deliver the benefits remotely.
Wow. So this research really opens up a potential new avenue, doesn't it? Using engineered cells, or maybe just their exosomes, to combat aging systemically, at least in primates.
It certainly looks that way, which leads to a really interesting thought to leave you with.
Go on.
If these exosomes are the main couriers delivering the anti-aging punch, could we potentially develop therapies using just these engineered exosomes? Could that achieve similar widespread rejuvenation without even needing the cell transplant itself?
Targeted delivery of rejuvenation signals without the cells. Definitely something to think about. Thanks for tuning in to this deep dive. Remember to like and subscribe if you found this interesting. It really helps us keep bringing you this content at the Sand Science Experience. We'll see you next time.